Hello, good morning, everyone. Let's get started. Today, we are going to be doing lecture 7, which is protein synthesis. Okay, first question. What is the central dogma of cell biology? This is something that we talked about before when we learned the cellular structures. The central dogma or the central theme of biology is that we start with information encoded in the DNA. And then we're going to copy a section of the DNA in the form of RNA. The RNA is then going to exit into the cytoplasm where it will be used. The instruction in the RNA is going to be decoded by the ribosome to make proteins. Okay, protein. So when you make a copy of DNA in the form of RNA, that is called transcription. Okay, transcription, and that takes place in the nucleus. Okay, in the nucleus. And then again, the RNA in the form of messenger RNA is going to exit into the cytoplasm where you will do translation. Okay, information on the RNA is used to make proteins. So that happens in the cytoplasm. It could either happen just with RNA, with ribosomes that are floating freely in the cytoplasm, or it could happen at the endoplasmic reticulum, the rough ER. Okay, so that's the central dogma of biology. Next, compare DNA. Sorry, I shouldn't have it off here. Compare DNA and RNA by completing the following chart. Okay, so this is more like a review. We learned this in lecture 2, but here it is. DNA. Is DNA single-stranded or double-stranded? You should know by now that DNA is double-stranded. Double-stranded. Okay, with a helix, right? Whereas RNA, it is just a single strand. Single-stranded. The type of ribose sugar in DNA, it is a deoxyribose. Okay, it's missing an oxygen, right? Compared to RNA. And then this one is the oxyribose. The possible nitrogenous base for DNA is C or A, C, G, and T. And for RNA, instead of having the thymine, you are going to have the uracil instead. So U is for uracil. As for the types of DNA, there's just DNA. Okay, there's not different form of DNA. But for RNA, there are 3 types. Does anybody know the 3 types of RNA? What are they? No? Okay, so we have the mRNA, the messenger RNA. We have the rRNA, and we have the tRNA. So those are the 3 types of RNA, and we're going to fill in their main functions right here. Okay, so first one, we will do mRNA, messenger RNA. Okay, probably should write a full name. The M stands for messenger. Okay. So what does the messenger RNA do? The main function of the messenger RNA is that it is a copy of the gene, copy of a gene, we can say copy of a gene, right? Which contains instruction to make proteins. Okay. So this is used by ribosome to build protein. Okay. So the messenger RNA is going to have these codes called codons. Each codon encodes for a specific amino acid. So the ribosome will come and then it will read the codes, and then it will know what amino acid to link together. Okay, so that essentially creates the primary structure of the protein. Next one, we have the tRNA. tRNA. What does the T stand for? Does anybody know what the T stands for? Okay, the T stands for transfer. Transfer RNA. Okay, so the transfer RNA looks something like this. Okay, it looks like kind of like a clover leaf, like that. Okay, and then it's a single strand. Okay, so the nucleotides here is going to bond with the nucleotides here to stabilize the structure using hydrogen bonds, of course. Okay, and at the bottom of your tRNA, there is going to be like 3 special nucleotide. Okay, so like, you know, U or something like that. So this is called an anticodon. And the anticodon will determine what kind of amino acid it is going to be carrying. Okay, so this is going to be an amino acid. Okay, so the tRNA is going to bring amino acids to the ribosome as the protein is being synthesized. Okay. The anticodon determines which amino acid the tRNA is carrying. Okay. Last but not least, we have the rRNA. rRNA. Okay, so this R stands for ribosomal. Okay. So the ribosomal RNA, it makes up part of the ribosome. Okay, which again is needed to build the protein. Okay. So if you draw it in like a picture, I guess, it will look something like this. So you have the messenger RNA that has the code on it. Okay, so this is the messenger RNA. And then you're gonna have the You're going to have the ribosome that's going to be clamped onto the RNA. Okay, this is, of course, very, very long. And you will have the transfer RNA. The transfer RNA that's going to be bringing in the amino acid to this machinery. Okay, and all the while, you're going to have a growing chain of protein. So this is the protein that's being built. The ribosome is made up of protein and rRNA. And then this thing here that's bringing in the amino acid, that's the transfer RNA. Okay, so the 3 different kinds of RNA is going to help with the synthesis of protein. Okay. Any questions? Okay, list the 3 steps of transcription. Okay, 3 steps of transcription. Transcription, again, is the process of making a copy of DNA in the form of RNA. So the first step is initiation. Okay. And then followed by elongation and termination. So in initiation, what happens is RNA polymerase, which is the enzyme that makes the RNA copy, binds to the promoter. Okay, the promoter of a gene. Okay, that marks the beginning of a gene. And then elongation is the actual building part. Okay, so we can say the template strand. Okay. RNA polymerase uses the template strand to build messenger RNA. And finally, termination. The process stops when RNA polymerase reaches the terminator sequence. Okay, which marks the end of a gene. Okay. So if you are unfamiliar with these terms, okay. So basically you have a gene here, gene, this whole thing is a gene. Okay. A gene is basically a segment of DNA that contains instruction to make a protein. And then at the beginning of the gene, we have a promoter sequence that marks the beginning. And then at the end of the gene, we have a terminator sequence. Okay. So promoter. Promoter marks the beginning of the gene. And then we have the terminator, which marks the end of the gene. Okay. Terminator is OR, yeah. The whole thing is a gene, and we're going to make a copy of that gene. in the form of RNA. Any questions? Can you shed more light on point 4? What do you mean point 4, Yusuf?
3 steps of transcription.
The what steps are the 2 steps? The 3 steps of transcription?
Yes.
Yeah, sure. Okay, let me bring up the PowerPoint then. Give me a second. Give me 1 second. Okay. So here, right, 3 steps, right? Initiation, elongation, and termination. So at the beginning, what's going to happen is the promoter, the RNA polymerase, which is the enzyme that makes the RNA, okay? When we do DNA replication, we have the DNA polymerase, right? So here we have the RNA polymerase, and it's going to have to bind to the gene. But the DNA is so long, right? Like this is the entire DNA, for example, right? Double-stranded. I'm just going to draw one strand, okay? So the RNA polymerase comes. How does it know where to bind to? Do I copy this section? Do I copy this section? Do I copy this section? Like where to go? Where to begin? Well, each gene is going to have a special sequence called a promoter sequence. Okay, so maybe you have a promoter here. Okay, maybe you have a promoter here. Okay, so it tells you where one gene begins and where the next gene begins. Okay, so the RNA polymerase will bind to the promoter. Okay, for this particular gene. Okay, so it's going to copy this particular section. So that's initiation. Okay, initiation. And then once you bind to that, then the RNA polymerase will open up the DNA strand, and then it's going to make a copy. Right, so as it's making a copy, it is called elongation. Now that's a little bit more complicated, so we can see in this diagram here. Okay, so this here, the blue is the parent DNA, and you can see there are 2 strands. Okay, one of the strand is called the template strand of DNA. And this is going to be very important for the later stuff that we're going to be doing. Okay, so the template strand is the actual strand that is used by the RNA polymerase to make your mRNA, to make your messenger RNA. Okay, the other strand that's not being used for transcription, that is the coding strand. The coding strand is the strand that actually contains the instruction to make the proteins. So you want to make an exact copy of the coding strand. Okay, in order for you to do that though, because of the complementary nature of how base pairing works, you have to use the template strand as your reference, as your guide. Okay, so think of this as like minting a coin, right? When you mint the coin, the mold that you use is going to be like a mirror image, right? An inverse of the actual coin, right? So that when you take it out of the mold, it's going to look like a normal coin, right? If the mold looks like what you want to make in the end, and then you mint the coin like that, then it's going to come out like inverted, right? It's going to come out backwards. Does that make any sense? So this is the template strand that we're going to use, and after you build it, it's going to be identical to the coding strand. And so this is going to continue until you get to
Thank you.
Okay, until you get to the terminated sequence, and then it's going to fall off. Okay, so that is the process of transcription. Okay, so what do you actually get when you do the transcription? Okay, you're going to get a messenger RNA, right? So we're going to draw and label the various part of the pre-RNA and the process mRNA. Okay, so here is let me try to do this Okay, so here is going to be your messenger RNA. Now, when you copy the messenger RNA, you are going to copy a bunch of stuff that, does not code for anything. Okay, so this here, the whole thing, this is called the pre-mRNA. Okay, pre-mRNA. Okay, so what do we have on the pre-mRNA? We're going to have these parts. They are called exons. Exons. Okay, so like you might have like exon number 1 here, exon number 2, exon number 3, exon number 4. So exons are the part that is being expressed. They are the coding part, coding portion of the gene. Okay. Now interspersed between the exons, you are going to have these introns. Okay, intron number 1, intron number 2, intron number 3. So introns are non-coding portion of the gene. Okay, so we're gonna remove the introns, okay, and glue together the exon. Okay, so here we're gonna have RNA splicing. Okay, in RNA splicing, introns are removed, exons are joined together. And you're going to end up having the final messenger RNA. Okay. Any question about intron and exon so far? Now this thing is going to break down very quickly if it goes out to the cytoplasm like this. So you're going to modify it a little bit, and you're going to add what we call a 5' cap. Okay, 5' cap. It's a guanine, modified guanine. You're going to add it to the 5', and then you're going to add what we call like a poly A tail. A, many A's. Okay. So this is called poly A, like adenine, right? Tail. Okay, so together, these things will stabilize the RNA. So all these things that are happening, the RNA splicing, the adding of the cap and the tail, all these things, we call these post-transcriptional modification. Okay, post-transcriptional modifications. Is that okay? I'm getting a warning on my screen saying my internet is unstable. If you cannot hear me, please do let me know. Or if I get dropped off, just stick around. I'll try to come back, okay? No question? Everybody's okay? Are you sure? Okay, yeah, thanks for letting me know. Yeah, if I'm lagging a lot, then and do maybe, you know what? Maybe I'll turn off the camera and see if that helps. You don't really need to see me, right? Okay, there. Okay. So if I'm lagging again, please do let me know. Okay, so now we have the table. Okay, this is a very important table, right? You have to know how to complete these tables. So we have a coding strand and we have a template strand.
Hi, Vincent.
Yes.
You are lagging seriously.
Like right now is very laggy. Yeah. Okay.
Seriously lagging.
Okay. Hold on. Let me try to go to another location. Okay. Hang in there, guys. Hang in there. Okay, now if I move to another location, I'm gonna lose my dual screen. So what that means is I cannot see the chat simultaneously on the screen. So if you have a question, like just unmute yourself and ask me, okay? All right, let me sit here. Let's see. Okay. Let me know if things are better, okay? If things are better now. Okay, so here, before we proceed, does everybody understand the difference between a coding strand and the template strand? And you have to understand that before we do these things. Any? No? Okay. All right. So let's talk about the difference here. Okay, so the coding strand, coding versus template strand. Okay, coding versus template strand. Let's say I have a DNA. Okay, DNA. So DNA is double helix, right? So that means I would have A, T, C, G, G, G. I'm just totally making this up. Okay, let's just say we have something like this on one strand. So what would it be on the other strand? Well, the other strand is going to be TAG, CCC, ATG, right? Okay, so this is just the DNA. Now, which of these actually codes for the proteins? Is it this strand or is it this strand? We don't really know, okay? So I will have to tell you in the question which one is the coding strand. So let's say this is the coding strand. Coding strand. Again, what does that even mean? Okay, coding strand is the strand that contains actual information, right? For making the protein. Okay. So this is the one that you want to copy. Okay. This is the one you want to copy. Okay. So we are going to open this strand up. ATC, GGG, TAC. And we're gonna open it up during the transcription process. Okay, so here comes the RNA polymerase. This is the RNA polymerase. RNA polymerase. Now, so it has two options. It's either going to build an mRNA using the top strand, or it's going to use it building using the bottom strand to build it. If it used the top strand, I'm going to end up having something that looks like the bottom, right? Do you understand that? Right? If I use the template, because they can only build by putting complementary base pairs, right? So if it used the top strand as a guide, then you would end up having UAG, CCC, AUG, which does not look like the coding instruction. Does that make sense?
Yes.
Whoever asked the question earlier.
Yeah, it makes sense.
Yeah, so that's why we have to use the other strand to guide the
What's the bottom strand called?
Sorry?
What's the bottom strand called? It's
the bottom strand is called the template strand.
Oh, okay, so you're mimicking, that's the template strand. And
That's
you're
right.
Top to get, okay.
That's right. So when you with the template strand, we will now be able to build it, right? Okay, GGG and then UAC. The main difference is that the T is replaced by the U, But the point is, if you use a template strand to build the messenger RNA, the instruction is going to be preserved. The instruction is same as the instruction in the coding strand.
Okay.
Okay.
Okay. Thank you, sir.
Yeah, no problem. So the key takeaway, key point here. Okay. Messenger RNA should be identical. Identical to the coding strand. Okay. Except like U replaces T. All right. Any other question before we move on? Okay. And I'm not lagging anymore, right? Don't
No,
think I
you're
am.
okay.
Okay, good. Okay, so let's go back to here. All right, so we have the coding strand here, and then we have we're gonna ask you to make the messenger RNA. Okay, so I just told you the messenger RNA should be identical to the coding strand, right? Identical. But you
Replace the U?
exactly replaces T.
Why do you do that? If you mind me asking. Sorry.
Why do I replace the T with the U?
Yes.
Yeah. So the RNA, if we go back to this chart, right? The difference between DNA and RNA is that there is no thymine in RNA. Okay.
Oh, okay. Yes.
Yeah. So that's why we have to use uracil instead.
Okay.
Okay. So go ahead, fill this in if you haven't already. I left my codon chart downstairs when I came up. I'm gonna go grab it. I will be right back. Okay, so over here, it's very simple. You just copy exactly what is shown in the coding strand. And I'm gonna replace all the U, all the T rather, with U's. So that's what you should have got. Okay. Now, anticodon, what is the anticodon? So again, if you have the mRNA here, mRNA, right, and then you have the AUG, AUA, CGA, blah, 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 okay? And then the ribosome is decoding this. Something needs to bring the amino acid over, right? Okay, so AUG is going to code for something. Okay, AUG, it's going to code for something. So we're going to go to a codon chart, and you can find the codon chart in the lecture notes. Okay, so the first base here is A, second base is U. Okay, so AU is this box. Okay, and then we look at the third base, G, right, which is right here. So AUG codes for this amino acid, MET, okay, which stands for methionine. Okay, so go back to our diagram here. So that means there is going to be a tRNA that's carrying the methionine. Okay, this is the tRNA. So how does the tRNA know to bring the methionine? Well, that's where the anticodon is going to come into play. the anticodon is going to be complementary to the codon on the mRNA. So that's going to be UAC. Okay, so this is anticodon. Okay, and this here, a triplet of nucleotide, that's the codon. Okay, so if the anticodon matches the codon in a complementary way, then the cell would know that this is the correct amino acid. Okay, now let's take a look at another one. Let's say we have another messenger RNA here, right? Okay, another tRNA. Okay, tRNA. And let's just say this one is UUU. Okay, UUU. Can someone tell me what amino acid this tRNA would be carrying? Okay, and you would need to have the codon chart. So I'm going to put the codon chart. Okay, now listen. This is UUU, the anticodon is UUU. So what amino acid would it be carrying? Okay, there we go. Okay. Maria said PHE, anyone else? Okay. Sienna also said PHE. And then LEU. Okay. Anyone else? Okay, this is a trick question. Okay. UUU, right? The anticodon. Okay. So on the mRNA, what would be the codon for this particular tRNA? What's the codon and the mRNA? That's right, it's AAA, right? So this is a codon chart, which means I have to look up AAA. So where's AAA? AA is this box, right? AA is this box, and then A is LYS. So this is carrying LYS. And then if this match, it will come. Is that okay? All right. If you don't fully understand, please watch the recordings. Where am I? Okay, so I was saying, right? I was saying, if I have this one, and now we know this is LYS, right? So this tRNA might come here, but then because UUU is not complementary to AUG, and so this particular amino acid is not going to come. Okay, the anticodon will only bind to the codon if they are complementary. Okay, having said that, if I have the mRNA here, then I can list all the anticodons, right? So all the anticodons would be complementary to the RNA codon. So it will be U, right? Because we don't have T. So UAC, UAU, GCU, UUA, CUA, AUU. Is that okay? All right. So amino acid now. Okay, let me copy this codon chart. I'm going to put it over here. It's probably too big. There we go. Okay, so we now have to look it up. Okay, we're gonna have to look up what the amino acid codes for. So we look at the mRNA, AUG. Right? So we come over here, AUG. So that's gonna be methionine, MET. Okay. What about the next one? Can someone tell me what the next one is, please? I want to make sure that everybody knows how to use this table. Okay, so what's the
Chile.
next? Yeah, ILE, right? Very good. That's isoleucine. And the next one? Can someone else tell me? What is the next one? Okay, how about this? Does anybody not know how to use the chart and need me to explain again? Please speak up now and then I will explain. I'm not sure how. Okay. So over here, right? Again, I cannot really see the chart at the chat. So if you post something and I haven't responded to you, just unmute. Okay. All right. So let's go through this. We are looking up CGA. Okay. CGA. So we find the first letter here. C. Okay. First base. Second base is G. Okay. And then you find out where these two boxes intersect, right? So it's here, right? This box is the box that I should be looking at. Okay. Does that make sense? The first step, right? So look up the first, look up the second, and then find out which box intersects. And then you have four options here, right? Are we talking about CGU, right? CGU, CGC, CGA, or CGG? Okay, it turns out they all code for the same thing, but we're looking for CGA, so it's this one. So that's going to be ARG, okay? Arginine. You don't have to know the full name, but it's just easier for me to say it. Okay, I hope that's better. Now, can you please do AAU? Okay, AAU. Please find out what AAU codes for. Now, you definitely need to have a copy of this chart for the test, okay? Otherwise, you're not going to be able to do it. Okay, so AAU is ASN. Very good. Very good. Okay, aspartic acid. And then next one, GAU. GAU is ASP. And then let's look up. I cannot provide it, right? Because like it's virtual, right? Like I cannot like give you a copy of it, right? Like you would have to, either have it on your phone screen or like, print a copy or something if possible. Yeah. Because I cannot provide it in a question. I think it's easier if you actually have a physical copy. So like you can write on it, right? That would be my advice. Anyway, can we look up the last one, the UAA? What is UAA? UAA. Yeah, UAA, if you look it up, it says stop, right? Stop. UAA stops, right? So that's it. That means you stop the transcription process. You stop the translation process. You stop making the protein. Okay. Now, why don't you take a moment and why don't you all take a moment and try the next one? Okay. This is a template strand. Okay. Is not the coding strand. Okay. So please be careful. I'm going to give you like maybe, a few minutes to try that. Okay. If you have questions, please do let me know. Okay. I'm going to go on mute and let you try that. Now, before we go too far, does anybody want to share their mRNA strand just so that I know everybody's in the right track before you like do the whole chart and then realize something is wrong? What is on the mRNA strand? Please, anybody. It's easier for you to unmute and read to me probably than to type it. Okay, maybe it's easier to type it. Thank you. So yes, very good. Is it Maria? Is that how to pronounce your name? I'm sorry if I'm pronouncing it wrong.
No, it's okay. It's Marisia.
Marisia. Okay, sorry with that. Thank you. So yeah, Marisia said this is AUG CCC GGU and then sorry GGA rather GGA and then UAU GCG and UAA. Okay, because the relationship between the template strand and the mRNA is that they are complementary, right? Complementary. Okay. Caramel asks, how do you get the T? Sorry, which T? How do you get the T? Can you explain to me which T you're referring to? Okay, maybe I'll come back to it. You can clarify in the chat box, Caramel. So that's that. Okay, anticodon now. So the anticodon is going to be, it is going to be, so I'm just looking for the blue color. It's going to be complementary to the mRNA, right? So it's going to be UAC, GGG, CCU, AUA, CGC, and then AUU. Okay. Finally, we're going to look at the amino acid, and we always use the codons, okay, not the anticodons. Right, so we're going to go ahead and use the chart to fill this in. So hopefully you will get MET, PRO, and stop. Okay, so you should know by now that it always begins with MET, right? Because MET is actually a start codon. So the start codon is AUG. Okay, it marks the beginning of the translation process. And the stop codon, there are 3 stop codons. Okay, it is UAG, UGA, and UAA. Okay, UGA, UAG. So in the lecture, I said, right, to help you remember this, and you know, you will want to remember it rather than look it up every time, is that, you know, you got A in biology, you are good, you are awesome. Okay, that's how you can remember it. Okay, maybe it'll help. So that's gonna mark the end of the translation process. Okay, now there's one more chart for you to do, and maybe you can use this for practice. Okay, so I will just you can check afterwards. I'm just gonna put it down. Okay, and you can check your answers afterwards because I'm gonna post this, right? Remember? So this is UAC. Okay. Now, I'm gonna put up like a, we'll call it like a cheating table. Cheating table. Okay, you probably want to have this cheating table near you on the test. Okay, So basically we have four things. We have the coding, we have the template, we have the anti-codon, right? Sorry, we'll say we have the codon, which is on the mRNA. And then we have the anti-codon, which is the tRNA. Okay, so basically, there are only four possibilities. So let's say we have ATCG, okay, on the coding strand. What would be the nucleotides on the template strand? Well, for coding and a template, they are complementary. So ATCG is just going to be TAGC. See, I'm still using the T and not the U, right? Because these things are DNA, right? Both of these. Okay. So for the codon, it is going to be same as the coding strand. So AUCG, but the U replaces the T. And then finally, you would have this. Okay. So the anti-codon is actually the same as the template. Okay, but U replaces T. So this is it. This is basically it. Okay. If you ever get confused or if you forget which one goes with what, this table should help you. Are we together? Anyone have any concerns? No concerns? Okay, let's see if I have any poll question for you today. Yeah, let's try this one. Go ahead. Okay, I only have 3 responses. Okay. The correct answer is this one, right? Because the DNA template here is going to be complementary to the RNA. Okay, so T is going to become A. A usually bonds with T, but because we're talking about RNA, it's going to be U, and then C, CGA, and then UU. Okay, when you get a question like this on the test, don't look at the answers. Okay, just write out what you think is correct first, and then match it with the answers. Okay, if you try to look at the answers, it's gonna for sure confuse you. Okay, so yeah, D is correct actually, right? Okay. Very good. Okay. Now, this is another one that you should probably think about. Okay. I'll let you try first and then I'll explain. All right. So we have answers kind of all over the place. Let's take a look at which one is which. So a codon, like normally in the body, has 3 nucleotides. Okay. So how many choices for the first nucleotide? Well, it could be A, C, G, or T. So there are 4 choices. And then 4 choices for the next one, because it could also be A, C, T, or G. And then also 4 choices. Right? So if you multiply these together, there are 64 possible combinations. So that's what normally happens. The question says if the codon contains 4 nucleotides instead of 3, so now you will have 4 times 4 times 4 times 4, okay? Or like 4 to the exponent 4, right, basically. So the answer is 256. Does that make sense of how I got the answer? No, it doesn't make sense. Okay, can you elaborate which one is the confusing part? Like, do
I
you
mean,
not?
it does make sense. I understand what you mean by like multiplying it 4 times 4 times 4 3 times. But I just don't understand. Like, if the question kind of confused me in a way.
Sorry, you understand why I multiply them together, but you're not sure why which part is confusing.
Well, mainly, okay, I don't. Okay, let's start over. I kind of don't understand the whole thing. If the coding contains 4 instead of 3, why you I don't get
Okay. Yeah. So do you understand what a codon is?
kind of.
Okay. So like, you know, the letter that we look up, like the AUG, when we look it up on that table, yeah, this is called codon, right? That's called codon. So a codon codes for an amino acid.
Yes.
Okay. So how many codons are there? Well, if you look at the chart, right, if you look at this table, right, and you count all the possible codons here. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, and you count all of them, there are a total of 64 codons.
Okay.
Okay, so why are there 64 codons? Well, the codon is made up of 3 separate nucleotides, right?
Yes.
So that's why we do 4 times 4 times 4. 4 times 4 is 16. 16 times 4 is 64.
Yes.
Right? So the question is saying, well, what if we have 4 nucleotides instead of 3, right? So blank, 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 right? So that's why I do 4 times 4 times 4 times 4.
Oh, okay. Okay.
Yeah.
Understand.
Okay. Good.
Thank you, sir.
No problem. Okay. Let's see if there's anything else we can do here. Yeah, we can do this one. This one is another one that we can do. Again, it's requires some thinking. Okay, please try it. Relaunch the poll. Go ahead. Okay. Everybody who answered chose C. So let's see if that makes sense. Okay, so the coding is AAA, right? And the anticodon on the tRNA, that's what we're looking for. Okay, so if the coding is AAA, so like you can use the cheating chart, right? If you want, right? If the coding is AAA, what is on the anticodon? Well, UUU, right? That's what it is. Right? So the answer is right here. Okay. If you want to think about it, then the coding is going to be same as the mRNA. Okay, so mRNA is going to be AAA, and then the anticodon will be complementary to the AAA, which gives you UUU. All right, moving on. Given the following mRNA sequences, which two codes for the same protein? Okay, so for this one, I mean, you just have to translate it, right? You just have to search up the code, and then you'll be able to see which two have the same amino acid. So, for example, you know, here is the table, and you know, you will look up AGU. Okay, so go ahead, look up AGU and see which amino acid it codes for. Okay, and if you go ahead and do it for all three of them, okay, you will see that the first one, you can do it later on your own time. Okay, the first one and the last one codes for the same protein. Okay. Okay, next one. More calculations. If a DNA sequence contains 360 nucleotides, how many amino acids do you expect to be in the protein sequence it encodes? Okay, so 360 nucleotides divided by 3, because 3 nucleotide makes up a code, right? Makes up a codon. So 360 divided by 3, it's 120. So answer is 120 amino acid, assuming there are no introns in the sequence. Remember, introns are supposed to be removed, right? So that's an assumption. Okay, moving on. Identify the type of mutation. Okay, so let me just quickly remind you what a mutation is. A mutation is basically, here, I'll do a summary here for you. For those of you who have not watched the recordings and not sure what a mutation is. Okay, so we have a mutation. Mutation is a permanent change. Permanent change in DNA. Okay, either caused by mistake during DNA replication, or it could be caused by mutagens. Okay, something that changed the DNA in the environment. So we have different types of mutations. We're going to talk about point mutation. Point mutation. Okay. So a point mutation could have a base substitution. Base substitution. Okay, you're replacing one nucleotide with another nucleotide. Okay, so I'm going to start with a very simple example, and I'm going to do it on the messenger RNA so it's easier for you to see. So let's say we have the original codon. Okay, let's say it is UUU. Can someone look up what UUU codes for, please, and tell me? Yeah, Marisha said PHE. Very good. So I'm going to do a mutation. All right. I am going to mutate the UUU. I'm going to mutate it into UUC. Okay. So please look up UUC on the codon chart and see what it codes for. And again, it codes for PHE, right? So what happens? After the mutation, even though I changed the codon, the amino acid did not change. So this is called a silent or neutral mutation. Okay, you wouldn't even know you have it, okay, because it doesn't do anything. Okay. Now remember the mutation. I am doing it in the codon because it's easier for you to process the information because you just look it up in the chart, right? But the mutation has to happen in the DNA. Okay, so in the DNA level, the code would have been like TTT, and then it got mutated to TTC. Okay, but when you translate the mutated code, it becomes UUU and UUC, right? Is that okay? Does everybody understand the neutral mutation before I go further with the other crazy stuff? It's just really hard to do this like virtually, to not be able to see students' faces. So I cannot tell from your expressions whether you understand or not. So no question. Okay, let's do another one. Okay, next one is called missense mutation. Missense mutation. Okay, so let's say you have UUU again, which codes for PHG, no surprise. We just looked it up. Let's say I mutate it to UUA. Okay, so please find UUA and tell me what UUA codes for. Thank you, Charles. UUA becomes LEU, right? Leucine. Right? So you see, it was supposed to be PHE, but now after the mutation, it becomes an other amino acid. This is a mistake. Okay, it's a missense mutation. Now, is this going to be bad for the protein? It could be bad. It depends where it happens. Do you remember when we talk about enzymes that I said enzyme has an active site, right? The active site is where you will fit the substrate in, right? To catalyze a reaction. So if this amino acid is in the active site and then it gets mutated to LEU, that could change the shape of the active site, and then the substrate would no longer fit. Right? So it could be bad, but sometimes it might not be bad. Okay, sometimes the amino acids are similar in structure or similar in properties that it's not gonna have a significant impact on the overall shape of the protein. Maybe the mutation it's not in the active site. Maybe it's just at the end of the proteins, and it doesn't really matter. Okay, so sometimes it matters, sometimes it doesn't really matter. It depends on the location, basically. Okay, so in the lecture I talked about sickle cell anemia, right, where a single point mutation in the hemoglobin protein causes the entire hemoglobin to misfold, causing your red cells to change into moon shape. Okay, so that is an example of missense mutation that have detrimental results. Okay, so let's try another one. Let's say we have UAC. Okay, please look up UAC, and then we are going to mutate it to UAG. Thank you, Marisha. UAC is TYR, okay, tyrosine. And then UAG is stop, okay? So this is called a nonsense mutation, nonsense. Okay, so let's say you have an mRNA and then you have a bunch of codons, okay? Blah, 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 blah. And then you have UAC and then blah, 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 like this. Okay, so suppose that's the mRNA. Okay, so you're gonna make the entire protein, right? But now you have a mutation, you change this to UAG. So when it's time for translation, you will make the protein, but all of a sudden you will stop over here because that's a stop codon and you will not make the rest of the protein. Okay, so this will lead to a truncated protein. A nonsense mutation is bad. Nonsense, S-E-N-S-E. A nonsense mutation is bad if it happens early on in the protein sequence. Okay, if it happens towards the end, maybe it's not as bad. So these are the three types of, base substitution. Okay, they're point mutation because it happens at a single point. Okay, so the other kind of point mutation is a deletion or insertion, and I'll show you what that means when we do the examples over here. Okay, so let's take a look and identify the type of mutation. So over here, this is the normal strand. So we have the codons here, we have the amino acids here, and let's take a look at the first one. Okay, so over here, we can see that the mutation occurs right here. Okay, right here. Okay, so instead of ACA, it becomes ACU. Okay, it's a base substitution, and the amino acid remains unchanged. Okay, so because no changes in the amino acid, this is going to be a neutral mutation, a silent mutation. Okay, no change. Okay, next one. The mutation happens right here. Instead of ACA, it's CCA now. And it encodes for proline instead of threonine. And so you have a different amino acid. We call this a missense mutation. Okay, and finally here, you have another mutation here that causes you to have a stop. Okay, there is actually like a double base substitution, right, with the U and the A here. And that causes a stop codon to occur prematurely. So that is a nonsense mutation. Okay, so the other situation here is a base pair insertion or deletion. That's when you like take out a nucleotide or if you put in a nucleotide. So you can see everything is the same up to the CCC here, CCC. And then what happens is after you take out the C, after you take out a C from here, okay, instead of reading the next codon ACA and UAA, now it is going to be like CCA, CAU, AA, and then you actually miss the stop. So everything downstream here is going to be extensive missense. Okay, and you're gonna miss your stop as well. Miss the stop codon. Okay, so by deleting one of the nucleotides, you will create what we call a frame shift mutation. Frame shift. Okay, because you're no longer reading it in the original intended order. Okay. So here, AUG, if you insert, this is an insertion. If you put in a U here, right? Supposed to be AUG AAC, but if you put a U right here, then you created a stop codon. Okay. So this also creates a frame shift. Okay. Resulting in a nonsense mutation. Okay. If you insert or delete multiples of 3, then there will be no frame shift, but you will end up, missing an amino acid, right? Okay, so deleting 3 nucleotides. No frame shift. Okay, but then you will be missing 1 amino acid. Now, is that bad? Again, it depends, right? Depends on where this amino acid is. It an essential one? Does it play a role in the active site? Contribute to structure? All that stuff matters. Any question before I move on? Okay, so maybe just take like a 5-minute break just to let you, freshen up a little bit, and then we'll do more afterwards. Okay, I'll see you in 5 minutes. The following is the template DNA of a section of a gene. Create a base substitution that results in a neutral mutation. Okay, so you don't have to write out the entire mRNA. I mean, you could if you want to, but for practical reason, let's say this is a question on a test, right? All you have to do is pick one. Okay, so we can pick this one, for example. Okay, so over here, TTA. Okay, so remember, this is template DNA. So on the mRNA, right, it is going to be AAU. Okay, AAU. So we want to get our codon chart for something like this. Put it right here. It's a bit too big. Okay, so right here. So I'm going to look up AAU here, okay, and see what AAU codes for. So AAU codes for ASN. ASN. So I want to create a neutral mutation, which means after the mutation, it should still code for ASN. Okay, so we can see here, that's ASN and that's also ASN. So if I mutate it from AAU to AAC, nothing changes. Okay, so I'm going to change this to C, which means on the template DNA, this is going to become a G. Okay, so that's one way of doing it. There are many, many possible answers, right? Okay. Is that okay? I'm just going to do another example, just in case that wasn't clear. So let's say I pick this one instead, GAG. So GAG on the mRNA is going to be CUC. So let's see what CUC codes for. CUC codes for LEU. Okay, and it looks like all of these codes for LEU. So I can change the last one to like a G. CUG also codes for LEU. No change. Okay, so which means on the template DNA, this is going to be a C. Okay, so that's how you create a neutral mutation. Any question about what I did there? No, no questions. Okay, next one. We have to create a base substitution that will result in a nonsense mutation. Okay, so it's worth writing it down here, right? The stop codon. Stop codons. Stop codon should be, you got A, you are good, you are awesome. Okay, so that's the stop codons. So we can write it out and see what it looks like on a template DNA. Okay, so on the template DNA, it should be complementary, right? Okay, complementary. So UGA, the stop codon UGA, should look like ACT on the template DNA. UAG should look like ATC, and then UAA should look like ATT. So that's what the stop codons should look like on the template DNA. So all you have to do now is scan through the template DNA and see which codon would become one of these if I change a single nucleotide, if I change a single letter. Okay. So for example, I have, let's see, I have a CTC here, CTC. Okay. So if I change the first one to A, then I get the ATC, which will create a stop codon here. Right. So that's one way of doing it. Okay. Is there an other way to do this? Can anybody identify another spot that I could change to create a nonsense mutation? I'm going to give you a moment to see if you can identify another location. Yes, very good. So AAT here, right? AAT, we can change the AAT to ATT, right? That would also work. Perfect. Moving on. Create an insertion mutation that results in a nonsense mutation. Okay, so this was a base substitution, meaning I changed a single letter. Now I'm going to insert something to create one of these stop codons. Okay, so I'm going to try to identify a location where after I insert a letter, it will become one of the stop codons. So I can do it, for example, like right here, I could put an A. Right? So when the ribosome reads it, it will be like A, TAC, CAG, GAG, AAT, CCT. And then instead of reading TTA next, if I insert an A, it will read ATT. Okay. And then ATT, of course, is stop. So it will stop. Okay. Now, sometimes people get a little bit confused. Okay. They're like, okay, I see A and T here. And then why don't I put a C here? Because ATC is also a stop codon, right? Okay, but that wouldn't work because when the ribosome reads it, it will read TAC, CAG, GAG. It will still read it as AAT, and then it will read CCT. Okay, so when you insert that extra nucleotide, you have to make sure that the reading frame will read that as a stop codon. Okay. There's another spot here. You can put an A here. That will work because AAT, and then it will read like ACT. Okay, so both of these would work. Is that okay? Like, I know it's a little bit confusing. Any questions from anyone? No? Okay, base substitution resulting in missense. That is very easy. Basically, for missense, if you change the first letter of anyone, it would almost guarantee to make a missense. Okay, so AAT, if you translate it to the messenger RNA, then it will be like UUA, and UUA codes for LUU. And if you change it to like GAT, then now it's going to be CUA. And CUA is going to be, oh, what is the chance of that? Okay, so that didn't work. We'll change the middle one then. Okay, we'll change the middle one. We'll change that to like C or some G or something. Okay, so then that would be UCA and UCA is SER. So that would be a missense mutation caused by a base substitution. Okay. All right, moving on. Now, this worksheet is one that you should definitely try on your own. I'll post the answer for all of it after class, so you can check your answers. But I'm just gonna do the first one for you, and then you can try the rest later. So this is the original DNA sequence, and the question says it is the template strand. Okay. So what happens is the mRNA sequence, I am going to do it as a complementary strand. Okay. Sometimes it might be useful to put little separator bars here to help you keep track of which one you're doing. Okay, because it can get a little bit confusing sometimes, especially if you're doing this on a screen. Okay. And then I'm going to look these up on the codon chart. Okay, I assume you know how to do that now. So if you look these up individually, you would get these amino acids. And stop. Okay, so that's the original. Right? That's without any changes, without any mutation. Okay, now there is a mutation. Okay, so I'm going to give you a moment to find out what is wrong here. Okay, when you think you have the answer, when you find out what the problem is, you can let me know. Okay. Okay, good. So Marisha said nonsense mutation in the second sequence, second codon. I believe that's what you meant. So let's take a look, right? We compare it base by base. So TAC, that is fine, no change. And then ACC becomes ATC. Okay, so there is your change. There is your base substitution. So what is the mRNA sequence? Well, mRNA sequence is going to be AUG and then UAG, and then the rest is the same. Okay, the rest is the same. What would the amino acid be? Well, UAG is going to be MET just like before, and UAG here is stop, right? So will this likely have an effect? Absolutely. Okay, what kind of mutation is this? This is a base substitution. Base substitution that results in a nonsense mutation. Okay, which creates a truncated protein. Truncated means shortened. Well, in this case, it's not so much truncated. It's like the whole protein is gone, right? Like you only have the amino acid MET, nothing else gets made, right? So is this going to be having an effect? Yeah, it's going to be very, very bad. You basically will not have that protein made anymore. Okay, so I encourage you to do all these on your own, and then I'll post the answer and you can check it after. All right. Long answer questions. Again, I will post these answers unless if there is one that you want me to explain. I kind of explained this one already, but you know I'll post the model answer for you to see. Here, I guess I can briefly explain. There are 64 codons, but only 20 amino acids. Explain why it is beneficial to have more codons than amino acids. So basically, the codons are redundant, right? You have more codes than amino acid. What that means is you have more than one code for the same amino acid. Okay, so why is this beneficial? Why is this a good thing? Well, this is a good thing to protect against mutation, right? So if you look at the codon, by having redundancy, like here, right? All these things codes for LEU, right? So, you know, if you have a mutation that goes from UUA to UUG, you're fine, you're okay. You're not going to have any detrimental results, right? And that's like an insurance policy. Yeah, if you only have one code for one amino acid, then all the changes, all the mutation will result in missense. Okay, and there's no protection. All right, so by having redundancy, it offers like a fail-safe a backup plan for mutation. Okay, deduce with reasons which process is being shown. Okay, so this one over here, you can see the green stuff that are coming out. Those are actually proteins being synthesized. Okay, so these green things, those are proteins. Okay, and then these are the magenta, purple kind of looking strand. Okay, that is the RNA, the messenger RNA. And then these lighter purple balls, those are the ribosome. Okay, so this is a translation. If I show you this picture and ask you what it is, translation. Anybody recognize what this is, the middle picture? What is that? This is what we learned last week. DNA replication. Okay, so you can see that there is a replication bubble here. That's a replication fork. You can't really see any of the proteins because they're too small. This one is transcription. Okay, you have your DNA here and all these things that are flying out. Right? These are the pre-mRNA. Okay. Next, we have, what do we have next? We have fill in the blanks. Okay, so let's do this. The process of copying segment of DNA into RNA is called transcription. The process of using mRNA to produce protein, it's translation. Okay, one follows the other. Something brings the correct amino acid to the ribosomes during protein synthesis. That is the tRNA. Post-transcriptional process involves the addition of a 5' cap, G cap, right, guanine cap, on the 5' end. And multiple adenines called the poly A tail. rRNA is the nucleic acid component of ribosome. The codon is a triplet of nucleotides found in transfer RNA. Oh, sorry, anticodon rather. Anticodon. And it is complementary to the codon on the messenger RNA. Start codon is AUG, which goes for MET. And then these are the stop codons. Number 8, the template strand is the strand used to create the messenger RNA during transcription, whereas the coding strand is the one that actually encodes for amino acid. During splicing of post-transcriptional modification, introns are removed while exons are connected. Mutations are permanent changes in DNA. Promoter marks the beginning of a gene. It is rich in adenine and thymine because, as you might recall, there are only 2 hydrogen bonds between adenine and thymine, but 3 hydrogen bonds between C and G. So it's easier to pull apart when you have more A's and T's. The terminator sequence marks the end of a gene. Insertion or deletion of nucleotides that are not multiple of 3 can result in frame shift mutation. And finally, a nonsense mutation results in premature termination of translation. So that is it for this tutorial. We do have a quiz coming up next Tuesday, okay? And the quiz will be on lecture 6 and lecture 7, okay? I will post some information about that after class. It'll be available, from 6 a.m. to 11:59, just like before. Okay, that's it. Well, thank you for coming. If you have any question, you can feel free to stick around or send me an email later on. Okay, have a good weekend.
Have a good weekend.
Bye-bye.